When we look at our living world, correct? So we know that all our organisms are interdependent on each other, so which is very, very nice. In any of the ecosystems, if you take, we know that organisms have a chain of dependency for energy, especially. We don't see that a plant depending upon an animal, correct, right? But instead, you see that an animal depending upon a plant. So, if you look at how we are surviving as organisms, one thing is for sure that plants are playing a very important role in getting this job done. Plants produce energy through the process of photosynthesis, which is very much known to us because we've been studying from a very small age. Now, this process of photosynthesis is not just any process where we are like, okay, this is a mechanical process or physiological process of photosynthesis, and we learn and we live it. It's not like that. It is more deep when you look at it. Because if you see, for the photosynthesis, what we have understood is the light is a very important source for photosynthesis. You see, so without the light, the photosynthesis will not happen, and the plants cannot know, or plants will not be able to understand how to get this job done in the absence of light. But you might argue there are other chemosynthetic organisms also, and they don't need they don't need the sunlight, but they are living. I agree with you. But if you see the amount of energy that is present, or the raw material for the energy that is present, solar energy is the highest, okay, renewable source of energy that we have for the production of energy in the biosphere, right? Whereas if you see the chemical or chemosynthesis process is quite um, less compared to that of the photosynthesis, right? So that is why we have to agree that light is very, very important source for the process of photosynthesis. So that means what? It says that in the absence of light, there is no life. It's simple as that. Now, what is happening here? We know that the light is used by our plants, and our plants, what they do is they undergo this biochemical process where they try to use the carbon dioxide and the light to manufacture something called as carbohydrates in the form of glucose, which in turn is stored as starch. Now, this stored starch, the plant also utilizes for its growth and development, as well as the other organisms that depend upon the photosynthetic organisms like plants. Herbivores. Herbivores eat these plants and they get some energy, and carnivores eat herbivores, and other carnivores eat other carnivores. So, like this, there is a chain of flow of energy in our environment. So if you knock out the sunlight out of the equation, then you can just conclude that it is impossible to sustain life without the absence of with the absence of sunlight, right? So now we will understand the process of photosynthesis. So now, if I ask you to write an equation on, okay, tell me how the photosynthesis happens. So now you guys are very smart, and I know that you've been studying for a very long time, and you derive an equation where you just tell that we need carbon dioxide, and then I need water, okay, and in the presence of sunlight, okay. Now what do I get? I get glucose, correct, and then I release oxygen. And this is a very basic formula that can represent photosynthesis process, correct, right? So this is the ultimate result, the end product of photosynthesis. Now, did humans just sit in a chair and then suddenly realize that, okay, photosynthesis happens like this? It happens to carbon dioxide, and then it requires water and sunlight is used for some process here, and then we finally get glucose. It did not happen like that, isn't it? So it was a very, very long story, and we have a very nice history to understand how the concept of photosynthesis was understood by us or human beings. Okay, so now you also have made some attempts to understand why oxygen is important or how oxygen is important, etc., etc. Let me remind you some of the experiments. So in the early experiments, so the next slide is actually pretty, what do you tell, congested, where a lot of facts are there. because the syllabus had to be completed fast. Anyways, so I will use this slide to, you know, share my lesson. So, anyways, what I was talking about, yeah. So when we talk about the process of photosynthesis, you might have understood by a little bit of experiments in the earlier classes. So let's start with one of them, which is very easy. So what you did, you took a leaf, right? You took a leaf, and then to this leaf, okay, now you try to put some plaster across the surface of the leaf, and then check for the presence of starch. So now you understood that the portion of the leaf that was exposed to the light, it showed positive for the starch, and the portion of the leaf that was covered by the tape, it did not show the presence of starch. See, the end result of the photosynthesis is to produce carbohydrates, and production of carbohydrates happens in the presence of sunlight. And one of the attempts that you did to understand was very simple. You took a leaf, you put a plaster on, and then you tried to test for the starch. You understood that the green portion, which was not covered by the plaster, okay, or the tape, was testing positive for the starch, whereas the other portion was negative. Okay, this was the first experiment, isn't it? And the other experiment that involved was, imagine that we have a leaf, and then we have KOH, no, KOH is potassium hydroxide. So we have a leaf and we have potassium hydroxide, and then we have a test tube. Okay. So let's say that we put this leaf in such a way, okay, that a portion of the leaf is within the tube and the portion of the leaf is outside the tube. And within the tube, what we do is we try to put some cottons that are dipped with KOH, or potassium hydroxide. Now, what is the speciality of potassium hydroxide? Potassium hydroxide takes away all the carbon dioxide. So when it takes away all the carbon dioxide, that means carbon dioxide is one of the raw materials for photosynthesis. And now what happens when you test for starch, the portion that was open with the air started, it was green and it was positive for the presence of starch. That means photosynthesis took place here. Whereas when you look at the other part of the leaf that was in the tube with the KOH dipped cotton buds, so what you find is you find that it tested negative. That means there was no photosynthesis happening. So one is the light, uh, and the other one is the presence of carbon dioxide, which you understood by simple experimentation. Now you might have understood that, okay, this is quite interesting. Knowing that, okay, this through these steps we can come to the proof. Proof is everything. Proof that, okay, these are essential. But let me tell you how the things unwound in the course of history to understand the process of photosynthesis. All right. So let's go back and see the history. So if you see the first attempts of learning photosynthesis, right? The first attempts of photosynthesis was learned by a very simple experiment. We call it as bell jar experiment. Okay. So this bell jar experiment, okay. So this bell jar experiment was conducted, okay. And this experiment, I think you already know about it. So now it's very simple. So what happened in the bell jar experiment was, if you see the, imagine that we have a candlelight, okay, and then we have a bell jar covering it. So now without the interaction with the air, now what will happen, this candle uses all the oxygen to burn, and then finally until the oxygen gets exhausted, the candle burns, and after that the candle actually goes off, correct, right? And imagine the same scenario, instead of candle we have a rat or a mice. So if I put a mice, okay, and then I close the jar, the mice eventually dies. So here also mice died, and here also the candle died. What's happening? So let us put both mice and candle in the same jar and then cover it. So here, in both the cases, actually the candle also burned out faster and the mice also died faster. And the third case, what are we doing? We will put a mint plant. So if I put a mint plant along with mice, candle, as well as with a jar, what happens here? Here, the candle does not go off, nor the mice dies. Both of them are alive. Now, how did it happen? Somehow, the mint plant was purifying something that was making the air toxic, right? So, so that was the first experiment that was conducted to understand the importance of oxygen or air. Okay, just give me a second. Okay, just give me a second, students. Uh, oh my, hold on. Right, 1 second, students. Okay, all right. Sorry, sorry about that. Okay, it was understood that somehow this mint plant is helping to purify the air. And this experiment was conducted by none other than Priestley, who is a very famous scientist who even was credited for the discovery of oxygen. Okay, so he conducted this experiment. Now, after he gave, after he gave his idea about how the photosynthesis happened, and then we have the next scientist, that is Egenhoff. So what Egenhoff does this, so it's given Jan Egenhoff, so it's von Egenhoff. So von Egenhoff, what he does is he does another set of experiment where he proved that sunlight is a very essential factor for the process of photosynthesis. So he showed that, like, yeah, the same thing. He showed that somehow something is happening between the plant and the candle as well as the mice. So that's what they were able to sustain. And he understood that somehow these respiring organisms like mice, as well as you take an example of a candle, they're disturbing the air. But whereas when you look at the experiments that was conducted by Egenhoff tried to prove that the light is very, very important, important part of the process of photosynthesis. Now, during his experimentation, what he did was he tried to grow some plants in the dark as well as in light. So in the dark, the plant did not test positive for the starch, whereas in the light, the plants tested positive for the starch. Next one comes the Shah's experiment, and the Shah says that okay, they are using light and they're using carbon dioxide. Okay, so now what I'm trying to tell is the whatever that energy that they're trying to prepare, it is in the form of starch. Okay, Shah says that the plants are storing food in the form of starch, and they're able to do it by manufacturing something called as glucose. Okay, finally we have another important scientist. His name is Eagleman. Okay, so Eagleman performs a very important experiment. What he does is he takes light and then he passes through a prism. Okay, so when he passes through the prism, you know that the light will disperse and it will create a spectrum. And in this spectrum, we know that we have Vibjor. Okay, so here we know that light has different wavelengths and it gives us the rainbow. Okay, the spectrum. Now he starts growing a blue-green algae. Okay, Chlamydomonas, and then he also leaves his bacterial phase. So algae and the bacteria were grown together. Now you know that the bacteria requires oxygen for surviving, and you know that this algae, blue-green algae, which is photosynthetic, requires light for survival. Correct, right? So now what happens is you can see that this particular algae, okay, started producing bubbles which were mostly in the red, and it is in the blue region. Okay, it is present in the red and blue region. So here it liberated more oxygen, and you can see that there was more bacterial growth. So this is the first time in first attempts in understanding, okay, that not all part of the light is used, only a certain portion of the light is used for the process of photosynthesis. So this was done with the experiments of Eagleman. Understood. And finally, we come to the conclusion. By the end of 19th century, see, all this effort started in 1760s, and it goes all the way till the 19th century. So by the end of 19th century, we had a clear equation. Okay, let's get back to what we studied. We studied that we had the first experiment, that was bell and jar, jar bell experiment with mice, mint, and candle. Okay, so after that, we understood that we have one more important scientist, that is Van Egenhoff. So what he did was he performs an experiment where he proves that light is important source. Yeah, his experimentation was he grew the algae in the presence of light. Okay, so in the presence of light, you could see that there were lots of bubbles, and then these bubbles was tested as oxygen. Okay, and then we had the next experiment of Shah's. Shah showed that the glucose is a byproduct of the photosynthesis, and it is stored in the form of starch. Okay. And finally, we have Eagleman. He split the light, and he showed that, okay, in the blue and the red region, the photosynthetic activity is very, very high using the blue-green algae and the bacteria. Clear? So finally, by the 19th century, we had this formula for expressing photosynthesis. Okay, so we know that 6 molecules of carbon dioxide and 12 molecules of water is required in the presence of light. Okay, what happens? Carbohydrate is formed, that is mostly represented by glucose, and 6 molecules of water and 6 molecules of oxygen is released. All right. So this is the equation that was done. And now you can see, hold on. Okay, so this was the equation. So I had one thing that I have wanted to point it out. Give me a second, students. Yeah, Van Neal is the person. So he basically tried to express this formula. Van Neal actually he gives this formula of 6 molecules of carbon dioxide plus 12 molecules of water that splits into glucose. That gives rise to glucose and water. See, what Van Neal does is, Van Neal is a very important scientist. Now, Van Neal tries to prove people that why water is an important source and how the sunlight is used. So he said that somehow, okay, somehow for the plants to be able to produce energy, that is your glucose, how is it doing this? So he said that we can use this carbon dioxide molecule. The carbon dioxide molecule can be reduced, okay, it can be reduced to form glucose. Okay, a carbon dioxide molecule can be reduced to form glucose. Now, how does this happen? If we find something that can oxidize this carbon dioxide, so now nothing, we have water, and we oxidize this water so that we get hydrogen and then oxygen. So this hydrogen is used for the reduction of your carbon dioxide and to fix it in the form of glucose. So that is why one meal's a contribution is very, very important. Understood? And now he also says that, okay, light does the role of splitting this water. It is oxidizing this water into hydrogen and oxygen. So now let us say that there is no water. What happens if we don't have light? So if we don't have light, that is when the chemoautotrophs comes. So these in chemoautotrophs which do not use light for the production of energy, they use the same carbon dioxide, but here we know that some of the molecules they use is in the form of sulfuric acid or nitric acid, so H2SO4 or HNO3. So here you can see these molecules are split, and the energy is used for the fixing or reducing the carbon dioxide to the glucose in chemoautotrophs. Understood? Is it clear? So what you have to understand, all the scientists and their experiments are very, very important, and their outcomes are very, very important. Is it clear? So now, in a generic sense, to understand photosynthesis, hold on. Students, tell me if you have understood till now. Can we go further? Yeah. All right. So next part we will discuss. One second. Yeah. Yeah, we'll talk about how the photosynthesis happens. So we know that in generally to describe the process of photosynthesis, the photosynthesis process usually 100% should happen in the presence of light. Okay. So that is fixed. Light is completely or 100% necessary for the process of photosynthesis to happen. That is fixed. Now, carbon dioxide molecules, which is present in the atmosphere, is fixed, okay, by using water molecules in the presence of light. So first, what happens is a photolysis reaction takes place where the water molecules are split using the sunlight. So here, the hydrogen atoms are used as electrons, okay, to fix the carbon dioxide, and the carbon dioxide is fixed as the carbohydrate, and finally the byproducts are water and oxygen. So here, all the green plants, they have to undergo this photosynthesis like this itself. There is no options, okay. Now, where does the photosynthesis happen? So now photosynthesis happens, we know that it occurs in the chlorophyll. Okay. Now we have that understanding. But it is said that the photosynthesis process happens in the greener parts of the plant. It's not just leaves, any green part of the plant, you can see that the photosynthesis is happening. Okay. Now, uh, the one of the major components or one of the major parts of this photosynthesis, the machinery where the photosynthesis happen, is in a cellular organelle called as chloroplast, which we already know. So chloroplast, which is a plastid, contains this photosynthetic pigments called as chlorophyll, and the chlorophyll is a very important pigment which helps us in photosynthesis. Correct, right? So now, talking about the the process how the photosynthesis happens is it starts with the beginning of trapping of light. So if you look at your chloroplast, hold on. If you look at the chloroplast is very well organized within the plant. Now, if you see the leaf, and if you take the cross section of the leaf, you know that leaf has mesophyll cells, right? So if you take the cell walls of the mesophyll cell, just very adjacent to the cell walls, the chloroplast are organized. Okay, very what you tell at the borders of your mesophyll cells, the chloroplasts are very well organized. The reason why they're almost aligned at the borders is because they want to be exposed to the sunlight as much as possible for the photosynthesis process. So now we know that the dorsal surface of the leaf contains more amount of the chlorophyll compared to that of the ventral surface. Okay. So now arrangement of this chloroplast is enabling for us to receive as much as light as possible. Correct, right? So now if you look at who is harvesting this light, so basically when you look at the spectrum of the light, we know that we have Webjaw within our spectrum. And in the visible spectrum, our photosynthesis takes place between 600 nanometer to 700 nanometer. That too, a very small portion of this light is used for the process of photosynthesis. Okay. So this wavelength of light is specifically picked up by a specialized pigments, which we call it as photosynthetic pigments. So these photosynthetic pigments are, any of these, pigments you see. So these are containing certain, certain chromogenic substances which are very sensitive to a specific wavelength of light. So what they do is, not all the light, to a certain portion of light, they're very sensitive and they know how to trap this light. Okay. And the example of the pigment you already know is chlorophyll A. Chlorophyll A is one example. Then we have chlorophyll B, and then we have xanthophyll. Okay, and then we have carotenoid pigments. So these are the pigments that are very important. Okay, so these pigments, what they do is they create this unit where they harvest the light for the process of photosynthesis. Okay, now coming to the chloroplast again. Now, what happens in chloroplast is a very important thing. We know that within the chloroplast we have the pigment containing we have the pigment that is chlorophyll and it harvests the light. So this is not the end of photosynthesis, students. So it is just the beginning. Okay, it's a part, only one portion of the photosynthesis where that light trapping process is happening. There's a lot of process that has to happen, and that goes with the structure of chloroplast. Yesterday we studied and understood the structure of chloroplast, but again we'll try to revise once again. So we know that the chloroplast is double membrane bond, And within the inner membrane of the chloroplast, okay, there is a uniform matrix and which we call it as stroma. Very, very important points. We'll take it up. So which we call it as stroma. And within the stroma, we know that there are like coin-like structures, which we call it as thylakoids, and the thylakoids are stacked upon one another to form, okay, to form something called as grana, right? So these thylakoids are interconnected by something called as stromal lamellae, correct? So these stromal lamellae, they interconnect each of the thylakoids. And we know that, understand this point, in each of these thylakoids, what do we have is the photosynthetic pigments. That is, the light harvesting pigments are present in your thylakoids. Understand this point, right? So this thylakoid region, and then you have your stromal lamellae. It is where the light harvesting systems are present, or the pigments are present, where they will do all the reactions which are corresponding to light, okay, or depending upon light. So we have sunlight, and whatever the reaction that is happening in the presence of sunlight is happening in the thylakoids and the stromal lamellae. Why? The reason is very simple. The light is harvested by the photosynthetic pigments, and these pigments are present there, as simple as that. After the light is harvested and we have the byproducts of the light reaction, so after light reaction happens, what do we get? We get the product, correct, right? So we get the product. Now this product is a raw material for the next set of reaction, and the next set of reaction happens in the stroma. So whatever the reaction that happens in the stroma, okay, do not require light, so we call them dark reactions, okay. So when I say light reactions, you can not conclude that okay, light reactions happens only in light and dark reactions happens only in dark, and they are not related. Definitely wrong, students. So we were just discussing three important parts of the chloroplast. One is your thylakoids, stromal lamellae, and stroma. Right? We have thylakoids and stroma, stromal lamellae. Here, the light reactions happens where the light is trapped. Once the light is trapped, and however the light is utilized, the byproducts of the light reactions are used by your stroma. Okay, this uniform space. So here in stroma, we have enzymes where these byproducts are raw material for some reaction called as dark reaction. Now, what is happening here? The stroma is able to do the dark reactions only on the products of light reaction. That means what? Dark reactions are light dependent. Understood? They are not independent. So they are dependent on each other. So that is why we should not confuse that light reactions are light, dark reactions are dark. No, dark reactions are depending upon the raw materials or the byproducts of the light reaction. Okay, where does light reaction takes place? It happens in the thylakoids and the stromal lamellae, and then we have dark reaction, and the dark reaction happens in the stroma. Correct? Okay. So this is about chloroplast, and we understood that a very important process is happening here in the chloroplast, where in one portion of the chloroplast, light is trapped and a certain biochemical process is happening, and the byproduct of that biochemical process goes to the stroma, and then stroma does undergoes one more process called as dark reactions, okay, and finally to give us the product. So all of them are interdependent, and this happens uh, you know this happens very regulated, okay. So it's very regulated. So now let us understand the pigments, and let us understand how the photosynthesis process takes place with a little more detail, okay. So now when we talk about the thylakoids, which are very, very important, so they are very important structural elements of the chloroplast. So these are flattened disc-like structures. We already know that. So each of this thylakoid, okay. So the thylakoid has a membrane. Yesterday I was pointing about the membrane of the thylakoid. So they are around 7 nanometer thick. So usually within the thylakoid membrane, you have something called as lumen, and this lumen of the thylakoid is around 5 to 70 nanometer width. Okay. So the thylakoid numbers can be up to 2 to 20 with respect to algae, okay, green algae, whereas in the higher plants, the thylakoid numbers are quite high, okay. So whereas when you talked about grana, which is the stack pile of thylakoids, so you know that up from 40 to 100 grana can be found in each of the chloroplasts. So that is the numbers are very, very high, correct, right? So let's see. Now coming to our thylakoids, as I told you, the thylakoid membranes, and then you have your chloroplasts that are present in the thylakoid membrane. Look at this. We have chlorophyll, we have carotenoids, we have cytochromes, quinones, and then ATP synthetase within the thylakoids. So what are they doing? They are very, very important for one of the photosystems or one of the light harvesting systems. Okay. So now let us understand the other important aspect of pigments. See, when I talk about pigments, as I told you, these pigments are of, you know, let's say that we have chlorophyll pigments, and let me point it out somewhere here. Okay, I will write here. We have chlorophyll A, and then we have chlorophyll B, and then we have xanthophyll pigments, and then we have carotenoid pigments, as we see here. Okay, we have carotenoid pigments, and then we have xanthophyll pigments. Now, look here, students. Now, what happens is we have a chlorophyll pigment, which is very, very important pigment. Okay. Now, let us assume that the chlorophyll pigment is here. Okay, this is the chlorophyll pigment. Now, forget about chlorophyll A. Apart from chlorophyll A, think that we have chlorophyll B, and then we have xanthophyll, and then we have carotenoid. Okay, we have carotenoid pigments. So all of these pigments, just imagine them as an antenna. Okay, so let me write an antenna. So we have an antenna. Okay, so here let's assume that we have chlorophyll B, and here we have xanthophyll, and then here we have carotenoid pigments. So what happens is when the light comes and incidents on the leaf surface, here these pigments, that is accessory pigments like chlorophyll B and xanthophyll and carotenoids are called as accessory pigments. So these accessory pigments, what they do is they increase the scope of absorption. So if I just tell photosynthesis, it's not one pigment, it's not chlorophyll. Chlorophyll can only do one part of a job. So we have other pigments, and these other pigments, what they do is they extend the scope of light absorption. So they will receive maximum wavelength of different wavelengths, and then they try to harvest this light. After harvesting the light, they transfer this light energy into to your chloroplast. So our chloroplast again uses this energy for the process of photosynthesis. So these accessory pigments together they combine to form something called as light harvesting system, or which call it as LHS. LHS we also call it as light harvesting system. Do we have it here? One second. Yeah. So yeah, these are the light harvesting. We'll talk about it. Okay, light harvesting system. So this LHC, which is a light harvesting system made up of carotenoids, okay, carotenes and xanthophylls. So what do they do? You can see here, carotenoids are basically what they do is they are not depending upon the light for biosynthesis. Okay, so they include two types. One is carotenes and xanthophylls. Okay, now when you talk about carotenes, you know that this carotene, for example, provitamin A is derived from carotene, okay, which is orange color pigments. Now xanthophyls are usually found in blue-green algae or the brown color pigments such as fucoxanthin, okay, and usually fucoxanthin, fucoerythrin are some of the xanthophyll pigments, and they are in matching with the ratio. If you see the green pigments and the brown pigments, they are almost in 2:1 ratio in the young plants. Okay. So these are the pigments. So now what these pigments together they do, they create something called as LHS or light harvesting system. So this light harvesting system, okay. So what they're doing, they absorb a lot of light. Okay. First thing, they increase the scope of this absorption. And the second thing, what they do is they protect your chlorophyll. So what do they do for your chlorophyll A? Your chlorophyll A, what it does is this particular chlorophyll A can become photo oxidized. Okay, it can become photo oxidized. What is photo oxidized, people? When there is a lot of light falling on your chlorophyll A, this chlorophyll A get oxidized, and this is not good for the chlorophyll A because it gets degenerated. So that's why what happens is this LHS or the light harvesting system or the antenna, it protects your chlorophyll A from getting photo oxidized. two functions. Okay, what is the first function? First function is to absorb broad range of light frequency, and second job is to protect our chlorophyll A from photo oxidation. Understood? Now here, apart from chlorophyll A, rest of the pigments, I told you that they create this antenna or light harvesting system. Now, what about your chlorophyll A? Our chlorophyll A alone can create something called as reaction center. Sorry about the slides. Okay, I know that it is quite disturbing without the pictures. Okay, so it creates something called as reaction center. So what are we talking about? We are talking about chlorophyll A. Chlorophyll A alone can create the reaction center. Means whatever the photosynthetic photolytic reactions that are happening, that is happening in chlorophyll A. So this chlorophyll A alone can create something called as reaction center. Now, this chlorophyll A is not same everywhere. We have two different types of chlorophyll A. Okay, hold on. Where is it? Yeah, PS1 and PS2. Hold on. Sorry, students. Okay, yeah. So I told you that each of this chlorophyll A can create its own reaction center. So based on the capacity of the chlorophyll A to observe a specific wavelength, we have two different types of photosystem. One is photosystem 1, other one is photosystem 2. Photosystem 1 was discovered first, photosystem 2 is discovered next. So that is why they are given 1 and 2, not the way they react in the photosynthesis. Okay, remember this. So now what is happening in photosynthesis 1, photosystem 1? If your chlorophyll A, okay, has the pigment, okay, anyways, it's a pigment. Our photosynthetic pigment is chlorophyll A. So if it absorbs around 700 nanometers of light, okay, so that is called as photosystem 1. Okay, so photosystem 1 of chlorophyll A, okay, basically chlorophyll A absorbs 680, sorry, I'll write it clearly, 680 nanometers of light. Sorry, 700 nanometers, I feel. Um, yeah, students, it is 700 nanometers. Yeah, so sorry, it is 700 nanometers. Okay, photosystem 1 is 700 nanometers, photosystem 2 is 680 nanometers of light they can absorb. So these photosystems, actually, what is a photosystem? It's a light harvesting unit. Okay, so it contains a reaction center, and the reaction center depends upon the chlorophyll A that we have. Chlorophyll A creates a reaction center, as I told you, right? So apart from chlorophyll A, we have accessory pigments also, such as chlorophyll B, carotenoids, and then which is less. So what they do, they absorb a light which is around P700. Okay. Now, if you look at the photosystem 1, okay, photosystem 1 and photosystem 2, they are present together in the thylakoids. Both the photosystems are there, whereas in the stroma only one photosystem are there. We will talk about it in detail, no problem. Whereas when it comes to photosystem 2, in photosystem 2 what happens, we have the same photosynthetic pigments such as chlorophyll A, B, carotenoids. Okay, now your chlorophyll A has the capacity to harvest only 680 nanometers of light. Okay, so that is why we have our two different photosystem, photosystem 1 and photosystem 2. So these light harvesting units, as I was talking, so this is how they are arranged. Okay, we have a reaction center of a chlorophyll A, and then we have the rest of the pigments. Now these photosystems, they collaborate together to perform the light harvesting functions. That means they will figure out how to take this light and use the light for the purpose of photosynthesis. Clear? Hold on. Are you guys understanding? Yes? No? Is the screen blur? No, it's not blur. Okay. All right, students, give me a second. So yeah. So next we have to understand what is light reactions and what is dark reactions. So anyways, we don't have enough time for that. So if you have any doubts, you can ask me. Okay. Or if not, you just let me know. Hold on. Where are you guys? Yeah, I think here. Yeah. If you have any doubts, please ask me, students. Okay. It's very easy. Okay. Let's not get confused. I know that the PPT was little not convenient, but next class I'll fix it. So for today, we understood very important concepts. Okay. And I hope that you remember these concepts. First of all, you remember the names of the experiments and the contributions of the scientists and what they discovered. So after that, we understood the very basics of photosynthesis, right? So that is, photosynthesis happens in the presence of water, carbon dioxide, and light, and what the equation is, and how the equation got derived. And next, we understood the structure of chloroplast and what is happening in the chloroplast and how the pigments are arranged within the chloroplast, hmm, and how they harvest light. Okay, light harvesting complex. If you have understood light harvesting complex and reaction center and the photosynthetic pigments, good enough for today. And finally, you just carry home this. Okay, in light reactions or in light harvesting systems, we have photosystem 1 and photosystem 2. And photosystem 1 and photosystem 2 depends upon the reaction center, that is your chlorophyll A. We have 700 nanometer one and we have 680 nanometer one. And together they form something called as light harvesting system, which we're going to study in tomorrow's class. Yeah. All right, students. Thank you very much. I will see you in tomorrow's class. Not tomorrow, Monday. Yeah, Monday's class. And we'll discuss further. All right. So if you don't have any doubts, you can leave the class. If you have, you can leave the message.